Hey there, guys. It's Mr. Kramer here, and I'm excited to present the number 3 protein synthesis transcription notes for you guys. While I'm gone today, it's important that you treat this video or this section of notes just like you would any section of notes as if I was there. We're going over some pretty important things, some of the things you may have learned last year, and then there may be some additional new things as well. So, as you guys know, whatever's in the red box, take the notes. The only thing that's different, there's quite a bit of practice in this section, so whenever there's practice, it's not in the red box. I would also put that in your notes as well. So with that said, let's get started. We have today's topic, and the date is on the board. Okay, so for our ICANN statements here, you can see the big check mark. We've labeled the 3 parts of a nucleotide and the hydrogen bonds that hold them together. We did a lot of that with our modeling activity. Then the 2 small check marks for ICANN statements 2 and 3 are simply because we've started those ICANN statements, we haven't finished them. So we have talked about the role of the different enzymes and replication, but we haven't done for transcription of mRNA, and we'll do that today. And then describe how complementary base pairing guides replication. We've done that as well, but not transcription translation. We're going to continue to work on that, along with ICANN statement 4 outline the flow of genetic information in cells from DNA to protein, including the processes of transcription translation, and then compare the structure of RNA with that of DNA. Okay, just before we begin the notes, I think it's important that we go ahead and do a small brain warm-up. So this is kind of a silly thing. You can try it yourself while you're watching the video. I'm going to put up 3 words. Say the color of the word and not the word itself. So here we go. How'd you do? Hopefully you were able to go green, yellow, red. Let's do the next one. Getting better? Did you say orange? Maybe you said orange. It happens. How about the next one? Like that's a super easy one. Red, blue, blue. Okay, next one. Kind of another easy one. But the last one maybe you won't see because this camera is covering it up. We can see it. Cool. Yellow, black, green. All right. Hopefully you did okay with that. Nice little way to get our eyes and our mouths and our brains all warmed up. So let's move on to our next slide and keep this brain warm up going. What I'd like for you to do is to practice writing out the base pairing rules. Let's suppose that we had a parent strand, like number 1 beginning with CAT. And what I'd like for you to do is to write underneath that the daughter strand. So pause the video and see if you can do it. Okay, so I've gone ahead and done the first one for you. I don't know. I think it's kind of silly that CAT also has GTA. It's kind of like a funny thing, like because cats often do Grand Theft Auto. Anyways, you can see I also divided the bases into groups of 3. There is a reason for that other than it's just simpler. It is a lot simpler, but those groups of 3 will be very important a little bit later in the unit. Let's pause the video and try number 2. I've already gone ahead and done the first 2 sets of 3 to get ATG and then CCC. I'll talk my way through the last one. So we know that C pairs with G, cytosine with guanine, and A pairs with T, adenine with thymine. Sorry for my really crappy handwriting. I'm doing one of the things where you write like with the mouse and you click. It's not easy. I'm not on my starboard today. C goes with G, and then our last set of 3 we have AAT. Good example of how hard it is to write. Look how bad those A's are. Good. Hopefully you understand these base pairing rules. They're very important and will really play a significant role in the remainder of the unit. If not, let's make sure that we do understand them before we move on. Okay, so what we're going to be learning today is we're going to be learning about how a section of DNA can code for a specific trait, or essentially to make a protein that if you make enough of those proteins, you'll get a specific trait. So if you can put enough proteins together, you can actually get a living organism, whether that codes for a living organism that's very athletic, or a living organism that's very smart, or a living organism that's very beautiful, or a living organism that is something, I'm not sure, famous, I don't know, I don't really know. I guess he was president, but yeah. Or maybe that section of DNA codes for something that's really serious. By the way, I took my camera off of this slide because I want you to be able to see this entire diagram. So let's say that we have a section of DNA, and this section of DNA is a real section of DNA that exists in your cells, in your DNA. And this section of DNA codes for a protein called hemoglobin. Hemoglobin is really important because it's the protein in red blood cells that carries and latches onto oxygen. So we have this section of DNA here, and we're looking at these three nucleotides, and we have C, T, C. And we know that that ends up pairing with G, A, G. Well, if we go through this whole process of creating a protein, the monomer for proteins are amino acids, and the amino acid that's needed in this place of the protein is glutamic acid. We're going to learn about glutamic acid and all these other amino acid names a little bit later in the week. But let's suppose that instead of an A, for some reason, instead of a T, this T here, we have an A. Maybe it was a mistake during replication, maybe it was some kind of damage that was caused externally, and so an A was kind of forced in its place. Who knows? But what we end up getting is, in the corresponding strand, we get a T here. And it should be an A. That's not good. That one small change puts in the wrong amino acid. This is valine instead of glutamic acid. And that completely changes the protein. We now have a mutant protein here. That one base pair reduces the life expectancy of a person from 78.7 years, which is the US average, to around 47 years. And it's the cause for the genetic disorder sickle cell anemia, or sickle cell disease. What ends up happening is hemoglobin has the incorrect shape, and that alters the entire shape of the red blood cells, and you get these kind of crescent or sickle shapes for red blood cells, and they can't properly carry oxygen. So people with sickle cell anemia have some very serious life-threatening issues that they have to live with. Some people have more severe sickle cell anemia than others, but it can be very, very dangerous. I mean, people with sickle cell, even people with sickle cell trait, have to be monitored while playing sports. People with the actual disease can't exercise normally. It can be very, very serious. So as we talk about, like, hey, this is the DNA and it's the code for life and it can make you beautiful or smart or athletic or whatever, that is true. But we also need to focus on one wrong base in the wrong place in a DNA sequence could completely alter a person's life as well. I turned my camera back on for this one. So those sections of DNA, they get a special name. They're called a gene. A gene is DNA instructions that control the production of proteins. And as we know, the instructions are in a code, and that code has 4 different letters that it can be in. It's not different than this code. Can you get that Rebus puzzle? I need to know, like, we need to do some Rebus puzzles while I'm there so we can give out some Smarties. So this would be head over heels in love. What about this one? That's an easy one. That's top secret. Speaking of DNA and code, I'm not going to show this video, but I will link it in the video tab, and you'd only need to watch like the first 2 or 3 minutes. This is a video from SciShow about how much of our DNA is really involved in the production of a protein. There was a study that came out 3 or 4 years ago that said 80% of our DNA is involved in the production of a protein, but the problem is only about 2% of our DNA is actually a gene that codes for a protein. So what's the other 78% do? It's kind of a weird thing. We've got a lot of DNA that scientists don't know exactly what it does. And so some studies say, well, they're like switches, and it turns off some genes and it turns off other genes, or it signals enzymes to bind and all this stuff. And then there are other studies that say not so much. So it's a really interesting thing. We've got a lot of DNA that doesn't really appear to do much, that's just kind of piled up in our genome throughout our evolution. It's cool. If you're interested in this kind of stuff, it's called junk DNA. You can watch the video on SciShow. It is a bit technical, but really cool at the same time. So again, I'll have the video embedded into the notes folder or linked in the notes folder, and you can click on it and watch it there. Okay, we're going to skip this because you guys are doing a video. So let's start with DNA versus RNA. I would suggest putting this T-chart into your notes, but the thing is, you already know this stuff about DNA. So maybe if you read that stuff and you feel comfortable, maybe you can completely avoid the T-chart. We do need to write these things about RNA though. We know that DNA's molecule is deoxyribose. Well, RNA's carbohydrate sugar molecule is ribose. That's why it's RNA instead of DNA. We know that DNA is double-stranded, RNA is single-stranded. We know that DNA contains thymine, RNA does not. Instead, it has something called uracil. And we know that DNA, there's only one type, DNA is DNA, but RNA, there are actually three types. There's mRNA, or messenger RNA, there is tRNA, or transfer RNA, and there's rRNA, which is ribosomal RNA. You need to know these, put them in your notes, pause the video, make sure you get them down. Here are the three types of RNA. Again, I don't have a camera because I wanted you to see the whole diagram. Though the two that we're going to talk about the most, and we're going to work with the most, are the messenger RNA. That's actually what this topic is about, making messenger RNA or mRNA. And we'll talk about transfer RNA. We don't talk much about ribosomal RNA. Ribosomal RNA is RNA that makes up parts of the ribosome. It doesn't ever go on to make a protein, so we don't really mention it. It gets made and it goes to the ribosome and forms ribosomes. But all three have very similar features. Again, they don't have thymine, they're all single-stranded, and they all have ribose instead of deoxyribose. If you want to see a similar diagram, you can on page 209 of the book. Okay, let's talk about how to make a protein. Making a protein is kind of like making anything. There's a recipe to follow. And so the first step is we need to make the mRNA from the DNA. Essentially, we're copying the instructions. And I like to make this analogy to baking a cake. And I can't bake cakes. I'm pretty bad at it. But you think of any valuable family recipe, there's probably that old recipe card that maybe your great-grandmother or your pro-yaya or whomever had that had to get copied down in order to make it through the family. For me, I know there's a cake that my mom makes. It's delicious. It's just called The Cake, and it has fruits, strawberries, and pineapple, and it's really wonderful. My mom got the recipe from her grandmother. And I remember my mom having an index card that she copied down my grandmother's recipe, which was on an older index card. Yeah, so it's kind of like just taking the recipe, the old recipe, and copying it down. So here I found online a recipe for a chocolate pound cake, and this is essentially what we're going to be making. But first we have to copy down that old recipe and put it onto something new. So the old recipe doesn't get damaged. That's step one. Okay, so the copying of the recipe, its actual scientific name is transcription. And so transcription's definition is production of mRNA copy from the DNA template. It's going to happen in the nucleus, something that's good to know. So the DNA is in the nucleus. It doesn't need to leave the nucleus. There's no chance of it going out and getting damaged. It just stays in the nucleus. There's a section of DNA, it could be a couple hundred bases, that are going to say, hey, yo, we start transcription here. That's called the promoter. The promoter is that section of DNA that says transcription begins here, and it's going to continue through the DNA to make the mRNA, and then there will be another set of bases a couple hundred or so, called the terminator. So essentially that is telling the enzymes, start here, make your mRNA, end here. And I just think it's funny that it's called the promoter and terminator. I think those are so aggressive terms, but I will go with it. Hydrogen bonds in the DNA are broken again by helicase, and then we have a new enzyme, RNA polymerase. It's going to read the bases on the DNA, on one side of the DNA, so it could read either side depending on which copy it wants to make. It's going to read the bases on one side of the DNA strand, and it's going to put in mRNA nucleotides to match the DNA bases. So, if on the DNA it says C, it's going to put in a G into the mRNA. If it says T, it's going to put in an A, just like our base pairing rules. But if it reads an A, it's not going to put in a T, it's going to put in a U. And we'll do some practice here in a little bit. You can see here in this diagram we have kind of like a replication fork again opening up. And you can see this red line is the mRNA that has been created from the template strand. After this step, we have our recipe. We're ready to go make a cake. Here we have our nucleus, and here is our another diagram. Here's our chromosome with our DNA, and we can see again, here's our enzymes that are our RNA polymerase that is making the mRNA. And notice that when there are A's, it puts in U's. There is no thymine in mRNA, it's uracil. Everything else is the same. When there's a C, it puts in G. When there's a G, it puts in a C. When there's a T, it puts in an A. Only when it would be a T, a U goes in its place. So the base pairing rules are slightly different for mRNA. This video is very, very important. I will also link this video or embed it into the notes folder. You absolutely should watch this one. This shows a computer animation of the transcription process based on real data. So watch this video. It's only like 3 minutes long. It shows you how fast it happens. It's something that you definitely need to watch. So stop this video, pause it, exit out, go to the other video, the one from the Dolan DNA Learning Center, and watch that video, and then come back to this one. Cool. All right, let's do a little bit of practice. Let's do some transcription. We have this section of DNA. We have CGA. So C we know pairs with G, G pairs with C, A pairs with U. And then we keep going. G, A, U, C, C, G, G, U, A. Gua. I want you to go ahead and try the second one on your own. And I'm going to pause the video and then we'll see how that goes. Oh, you should pause the video too. Sorry, I said I'm going to pause the video. You pause the video and then we'll see how it goes. Here's our first strand. And then if you're looking for the second strand, hopefully you were able to get AUG, ACG, UUU, AGC. You now can do more of the transcription and translation worksheet. You should be able to do all the way up to this step right here. So we're not quite to translation yet, which is going to happen down here. So you can't do this yet, but you can do both of these for all of the problems. Last but not least, instead of a normal summary, we're going to do a summary quiz. And then the last slide will be the answer. So here's our first summary question. Just go ahead and write these down in the summary box. I believe there are 6 questions. The first question says, which of the following is not true of RNA? A, it contains ribose sugar. B, it is double-stranded. C, it contains nitrogen-based uracil. Or D, there are multiple types. If you need more time, go ahead and press pause. Moving on to number 2, which of the following in the list below is not a type of RNA? A, tRNA, B, mRNA, C, nRNA, or D, rRNA. Again, if you need more time, go ahead and press pause. Number 3, which of the following DNA nitrogen bases are replaced by uracil in RNA? Is it A, adenine, B, thymine, C, guanine, or D, cytosine? Pause if you need more time. Number 4, which of the following is the name of the process in which a copy of DNA, it should be is made to create a protein? A, replication, B, transcription, C, translation, or D, synthesis? Pause if you need more time. If you sound like a broken record. Number 5, where in the cell does transcription take place? A, the nucleus, B, the ribosome, C, the mitochondria, or D, the cell membrane? If you need more time, pause, but we're on to number 6, which is the last one. So far, how do you feel about your knowledge of DNA and the beginning of the protein synthesis process? A, you're like a 1, like I don't know anything and this is difficult. B, you're like a 2, you're lost, but you could get there if you pay a little bit more attention. C, you're okay and you're getting better with the learning objectives. Or D, you could probably teach a college class on this stuff. You're dominating. You're feeling really good about it. Obviously, number 6 doesn't have an answer, but for the other 5, you should have got B, C, B, B, A. If you didn't, it could be worse. You're not Wayne Rooney in this picture. He literally just jumped on his head. I'm not sure why. I'm not good at soccer, but I don't think that's how you're supposed to play. Guys, you can work on your booklet once you finish this. I hope you have a wonderful, wonderful day, and I will see you next class.